Hello, welcome to the National Center of Forensics webinar series. I will be presenting the speaker for the DNA mixtures webinar, and her name is Amanda Baker. Amanda Baker is a forensic examiner in the DNA casework unit of the FBI laboratory. She started her career with the lab in November of 2007. Mrs. Baker has held multiple positions within all DNA units from January 2009 to March 2015. She was a biologist within the DNA unit, processing items for evidence for serology and DNA analysis. Since March 2015, Mrs. Baker has been a DNA forensic examiner and is qualified in DNA analysis to include statistics using probabilistic genotyping. She has been qualified as an expert and has testified both in state and federal courts over the 20 times. I present to you Miss Amanda Baker.
Thank you so much, Jasmine. Hello, everyone, and thank you so much for taking the time to watch my webinar today. I'm hoping to give you a little more information on DNA and DNA mixtures and how we can use them forensically. So again, my name is Amanda Baker. I am a forensic examiner in the DNA casework unit at the FBI laboratory. The laboratory is located in Quantico, Virginia. So first and foremost, just our little spiel at the beginning. Any names that I use of any manufacturers for providing are provided for identification purposes only, and inclusion does not imply endorsement of that manufacturer or its products or services by the FBI. The views expressed today are those of myself and do not necessarily reflect the official policy or position of the FBI lab or the government. So today what I hope to do in the next hour or so is to give a pretty brief rundown on what DNA is and how it's unique to individuals, how forensic DNA laboratories can utilize DNA, learn about DNA interpretation and comparisons very surface level, and then hopefully give a little more understanding on mixtures and probabilistic genotyping. So to begin, what is DNA? So DNA stands for deoxyribonucleic acid, and it's our genetic makeup. You can think of it like our body's blueprint. It gives instructions on how we look as well as how our body functions. We each have two copies of DNA. One we inherit from our mom and one we inherit from our dad. And all of us have the same sections of DNA, but what differs is the repeats of that DNA, which we'll talk about soon. Those repeats is what is inherited from our parents. Over 99% of our DNA is the same, which makes us all human, and that all makes sense. Most of our friends and family, we all have arms, legs, heads, shoulders. We don't have tails and we don't have scales. With the exception of identical twins. So identical twins do share all of their DNA. So if I were to type their DNA in the laboratory, I would not be able to tell the difference between identical twins. Fraternal twins do come from different eggs, so they do have different DNA. A small portion, less than 1% of our DNA, does differ amongst us, and really what we focus on for forensic purposes. And then we also have sex chromosomes, which make us biologically males or females. Females have two X chromosomes, and males have an X and a Y. DNA is found in virtually every cell of our body. So if you think of a cell like an egg, the yolk of that egg is the nucleus of the cell, and that's where our DNA is found. We mostly focus on nuclear DNA. There are different types of DNA that we'll talk about in a few minutes, but nuclear is most discriminating. So that is what we inherit, half from mom, half from dad, makes us all slightly different. There are some other DNAs within the body that are the same as some of our parents or our offspring, and we'll get to that in a second. Our body has trillions of cells, so most every cell has nuclear DNA, and it will be the same across cells. And it doesn't matter where it's collected from. So whether it's a blood sample, a saliva sample, a semen sample, I would expect the DNA to be the same across all of those. And it's the same throughout our life. So from birth to death, our DNA is the same. Are there different types of DNA testing? I've alluded to that. Yes, there are. So again, the first is nuclear. So this is the most discriminating. We inherit half from mom, half from dad. A majority of our cases undergo nuclear DNA testing because it is the most informative, and we're focusing on non coding areas. I'm sure you've heard of genetic genealogy and building family trees or ancestry. While that is types of DNA testing, we do not use that currently for forensics. So we're actually looking at non-coding areas. So the areas we're looking at, it doesn't give a person their hair color or their eye color. It's essentially just a bunch of letters that really don't mean anything. We also have Y chromosome testing. So Y-DNA is passed down along the paternal line from father to son. So my father-in-law, my husband, and my two boys all have the same Y DNA. If I have any future grandsons, they will also have the same Y-DNA as my two children. So you can see here, I have a little bit of a tree for you. So the top green square is dad, his male offspring, and the next line, the two green boxes have the same DNA as he does. Um, any males that his daughters may marry potentially will not have the same Y-DNA. That will come from a different line. A lot of times we use Y-DNA testing in sexual assaults where we have a lot of female DNA, for example, on a vaginal swab. We want to target potentially that low-level minor contributor, that person that left behind a really small amount of DNA. We may be able to essentially ignore the nuclear DNA and we will look at the Y-DNA. And then we also have mitochondrial DNA that's passed down from mother to child unchanged. So my mom, myself, and my two children all have the same mitochondrial DNA. However, since I do have two sons, they will not pass on their mitochondrial DNA to any future grandchildren I have. They will inherit that from their mother. So my daughter-in-law in the future will give her mitochondrial DNA to any of her children. So again, here's an example. Grandmother is the orange circle at the top. She's passed it down to all of her, all of her offspring on the second line, and then the female offsprings pass it down to the third generation. This a lot of times we use this for cases where we have hairs or bones, bodies that have been buried out in the desert. Usually, the nuclear DNA is no longer working; it's degraded, it's cut into little pieces, and we can't get a full nuclear DNA profile. So we will look at mitochondrial DNA. There is a lot more copies per cell, so it doesn't degrade and break down nearly as fast as nuclear DNA does. So what are we testing for in DNA? We're testing for something called short tandem repeats, or STRs. And these are small pieces of DNA that repeat themselves over and over again. We all have those same sections of DNA, but what makes us different from each other is how many times those sections repeat. So, for example, let's say my dad, at one small section, my dad gave me 10 repeats of that location, and my mom gave me 15. My DNA type at that location would be a 1015. Now, I could have a friend or another family member who may inherit 9 repeats from dad and 12 from mom. Their DNA type would be a 912. Where do we get DNA from? The short answer, just about everything. We can get DNA from especially body fluids, blood, saliva, semen stains. Those are really good sources of DNA. But then also anything that's been worn or handled a lot. Things that are very porous, very rough. They can retain some of the skin cells that are taken off from your hand to that item. We would expect to get DNA there. Anything that comes in contact with the mouth, cigarette butts, bottles, cups, gum, those are also really good sources of DNA. Touch DNA at the very bottom of this slide, I do say yes and no. So touch DNA, again, anything that's handled a lot or worn a lot, clothing, firearms, baseball bats, I would expect there to be a lot of transfer from a person's skin to that item. Touch DNA that we don't get a lot of DNA from, we call this low copy number. And really, it's having a small amount of starting cells. So if I were to touch a pen, for example, really quickly, I may or may not leave a few cells behind, and there's not enough there for me to really detect the DNA. So there is a such thing as not enough DNA to detect. But again, the more an item is worn or handled, the more I would expect to obtain a pretty decent DNA profile from. So the first thing we typically do in the lab is forensic serology, and this is a screening tool that we use to identify body fluids such as blood and semen. We know these areas are rich in DNA, so it helps us with our DNA testing. If we have a sexual assault that occurs on a king size comforter, a king size mattress, it would take a really long time to take essentially a Q-tip and swab that entire mattress to try to recover any DNA. It would take a lot of time, it would take a lot of resources. So, knowing in the example of a sexual assault, there may be blood or semen deposited on that mattress or that sheet, we will look and see if we can find those stains first and then start there for DNA, knowing it will only take a really small amount probably to get a DNA profile. What is a DNA profile? So, a DNA profile is the visual representation of an individual's DNA types. We call this graph an electropherogram or an egram for short. You can think of it like spikes on a heart monitor. So, it's a graph with peaks, and each of those peaks has a number, and that number represents the amount of repeats of that section of DNA. Ideally, in an ideal world, I would see one or two peaks at each location. So, again, one from mom, one from dad. If I only see one, it could be that actually mom and dad gave me the same number of repeats. So, mom gave me 14 and dad also gave me 14. So, I would only see one 14 peak there. Again, in an ideal world, I would expect both of these peaks to be of even heights. So this is very important later on when we're talking about how to interpret a profile and determine how many number of contributors there are. Keep that in mind that I would expect there to be some balance between those two peaks. Um, we look at 21 locations on an individual's DNA, or 21 of those short tandem repeats. In addition, 3 sex typing. So that lets me know if I have a biological female sample or a biological male sample. Amylogenin is the marker that I always see. So males have an X and a Y, and females have two X's. Then I also look at Y-indel and the Y and a YSTR locus. That's only for males. So if I see a peak at those locations, it lets me know that a male is present. So here's an example of this. So each of those green bars at the top, the green rectangles, that represents one of those STR locations. So there are 21 of them. The purple circle to the left, that is Y-indel, that is only present in males. The blue circle is amylogenin. This is where I would determine if I have two X's or an X and a Y, so I can tell if it's male or female. And then the red circle at the far right, this is one of the YSTR loci that, again, if I see a peak there, that lets me know that I have male DNA. Our DNA is then separated by size to give me each of those individual sections. So that's why it's broken down into those individual rectangles. This is an example of what a male profile looks like. So in the red circle to the left, you see a peak that has a number 2 in the box under Y-indel. There's also an X and a Y under the AMEL, the amylogenin box. And then there's also a peak to the far right under DYS 391. Using these three pieces, I can determine this is a male profile. I do not need to have information at all three. However, it is possible if a sample is degraded or broken down, I may only have information at one of the three, but I could still determine that it's male. A female profile. So here in the red circles, both to the left and the right, the far outside boxes, you see no peaks. And then for amylogenin, you see one single X peak. This lets me know that this is a female. There is no Y peak next to that X, and there are no peaks at the two Y loci. So this is what a female profile would be. And also, again, looking, there's 1 to 2 peaks at each of those green rectangles, and they're all relatively even in height. So I know this is a single source profile, meaning from one individual. So, how do we develop a DNA profile? In the laboratory, it's a step by step process. The first step is collection. And this is where we're going to take a Q tip or a swab and swab an item or take a cutting of that item. And what we're trying to do is take a portion of the cells that are on that item so that we can get the DNA from the cells. So that's the first step, collection. The cutting or swabbing is then placed into a tube. It could also be a liquid sample if it's saliva or blood, for example. The next step is then extraction or purification. And this is where we're going to add chemicals to that tube to break open those cells. So if you think of my earlier example of a cell being like an egg, essentially what we're doing is we're cracking the egg and we want to wash away everything we don't need. The white of the egg, the shell of the egg. We really want to focus on the cells that are in that nucleus or in the yolk of that egg. So the extraction process does just that. It opens the cells and it cleans the DNA. This is done via robotics. It's pretty hands off. Once the biologist places the chemicals in the tube then goes onto this instrument, and the instrument does this purification for us. Batches and controls that I have here. So we do process samples in batches. So I could have multiple items from one case or multiple cases on one batch. And because of that, we have controls. We have a positive control and a negative control. A positive control is a known DNA sample that after it goes through the entire laboratory process, we know what the answer should be. So we're going to compare that to make sure that our instruments and our reagents are working appropriately. We also have a negative control, and this is a sample that does not have any DNA. And at the end of the entire process, we hope it still doesn't have DNA. This is going to make sure, again, that there isn't something weird going on with our instruments. We're not getting cross-contamination. There's no contamination in our reagents. So because we use robotics, batches and controls are very, very important to use. The next step, excuse me, is quantification. And gives us an estimate of how much DNA we've recovered from that collection. So from that swabbing or that cutting, it lets us know how much DNA we expect to have there in our sample. It's also our first indication if we have male DNA present. And again, just with extraction, we also process in batches and we also use controls since this is mostly robotic and hands-off. The fourth step is amplification. You can think of this like genetic photocopying. Again, same as the other two steps, robotics, controls, and batches, very important. So this is taking the DNA and making millions and millions of copies of it so we can essentially, quote unquote, see the DNA, which is the separation phase. Again, robotics, batches, and controls are very important. This is where the DNA is going to be separated by size so that I get that ultimate electrophoregram or those peaks on the graph. So then after DNA profile is developed, it's then my job as the forensic examiner to interpret a profile. So how do I do this? First, the easiest thing to do is determine, is it male or is it female, or a mixture of both? So I'm going to look at those three sex determining areas and determine, do I have male, female, a mixture of both? I'm also going to determine number of contributors. Do I think it's one person, two, three, four, etc.? So again, if I have two peaks at each location, it might be a good indication that I only have one individual present. However, at one of these red bars, for example, if I see 3 or 4 peaks, that may give me an indication that I have more than one individual. And then there are some biological artifacts that we can get just because of the DNA copying process. We call this stutter. So we account for that in our standard operating procedures based on validation. We have determined how often we expect stutter to appear and at what level it will appear. So I wouldn't expect a stutter peak to be as high as a regular allelic peak. So we have percentages that we can take into account to determine, is it truly a person that I'm seeing or one of these artifacts? Same thing with bacteria. While our DNA kits are human specific, there are some bacterial DNA that will sneak in there every once in a while. And again, we have seen this in validation and can use that for our interpretation using our standard operating procedures. So a single source profile, I've talked about this before. I would see 0, 1, or 2 peaks at each location. It's going to depend on the individual that I'm collecting that DNA from, whether they have 0, 1, or 2, and the amount of DNA present. If I only have a few starting cells, I may not see information at every one of these 21 locations. If I have a lot of starting DNA, more likely I'm going to see DNA at each of those locations. Again, we talked about this. A DNA profile is peaks on a graph. It doesn't give me any identifying information except if an individual is biologically a male or a female. So, with that being said, it's very important that we have reference samples. So, a reference sample is a sample taken directly from an individual, whether it be via a buccal swab or a cheek swab, a blood sample, or some other alternate known, a hairbrush, for example, or a toothbrush. Something that I know when I develop a DNA profile, it belongs to that individual. So if I were to swab my cheek, process that swab in the lab, the DNA profile at the end, I know belongs to me. We refer to this as a known, or you may hear me say a K. And this, I'm expecting it to be single source, 0, 1, or 2 peaks at each location. It should almost always be a full profile. Typically, blood samples, saliva samples are rich in DNA. And we use this known to compare to any DNA we may get from an evidence sample being an item which we don't know if DNA is present, and if so, who that DNA may belong to. So after all of that, we've developed a DNA profile. Now what do? We perform comparisons. So here's an example of a single source male contributor. This, let's say, is a swabbing from a water bottle, for example. And then on the right-hand side, I received a known from a male and a known from a female. And what I can do is I can compare those numbers at each location to see if an individual is included or not. So if you were to look at the upper left bar that says D3S1358, there is a 15 peak and a 16 peak. The male K also has a 1516 at D3. The female has a 1618. Those are not the same, so I can say that that female is excluded as being a contributor to this sample. For the male, those first two match, but I do need to compare across the entire profile to determine that, yes, these two are consistent and similar, and I may have a possible match. What are my results of comparison? We just talked about this. So an exclusion means that there are differences between the question sample and the known sample. This means that that known individual could not have left their DNA behind on that item of evidence. And then an inclusion means that every piece of information is the same, so that person could be a possible contributor to the evidence. So once I've determined that there is a match or a possible inclusion, what do I do now? Now I have to calculate statistics. And why do we need statistics? So we need statistics because not all matches are created equal. We know based on testing that a person's complete DNA profile of 21 locations is most certainly unique. However, I cannot say that with absolute certainty. This being, I cannot test every person in the world, everyone that has lived, or everyone that will live. But we expect each person to be unique, with the exception of identical twins. Not all evidence gives us a 21 locus profile. So if you think of a DNA profile like a phone number, sometimes we get equivalent of just the area code of the phone number. So if you were to take my area code 703, there are probably a million people that would match those 3 digits. As I add more digits on, fewer and fewer people are going to match until you get to my specific phone number. So we calculate statistics to show how great the match is based on the profile that we get. The more complete the DNA profile, the less likely the match could occur by chance, and the opposite is true. The less complete the profile, the more opportunities for a coincidental match. So again, if I tell you I saw that I missed a call and I only recognize the 703, it could be a plethora of individuals that called me. As I get more and more information, more digits of that phone number, I can narrow it down to potentially one single caller. So sometimes I know The feedback from the conference last year was talking about mixtures. Mixtures can be complicated. Unfortunately, we can't get super into detail in an hour time. My training did last 2 years, so it wasn't all about mixtures, but I spent quite a bit of time on mixtures. But hopefully, this second half of this presentation will give you a little um, uh, detail as to why mixtures maybe can be so complicated. So why is that the case? Well, first we have to define a mixture. A mixture is having more than one person's DNA type in a sample. So if my husband and I share a water bottle, I swab the mouth portion of that water bottle, I could pick up both of our DNA. It's going to be a mixture, having both of us on one sampling. A two-person mixture, this could be 0 to 4 peaks per location. So again, single source is 1 or 2, two-person is 1 to 4. This depends again on the person's DNA. It depends on how much sample is there to begin with, and it depends on sharing. So relatives share. So my brother and I, we share somewhere between 0 and 100% of our DNA. I have not typed either of us. I'm actually typed at the lab, but my brother is not, so I can't give you an exact percent. But it is possible because we both have the same biological parents that we are going to share some of our DNA. So that may look like it's a 1 or 2 person mixture depending on how much we share. When I say sharing, if you were to take a look at the second box from the left, VWA, there are 3 peaks here, a 14, a 20, and a 19. If I tell you this is a 2-person mixture, you would expect no more than 4 peaks. So it's possible that one of the contributors is a 1419 and the other one could be a 1420 or a 1920 or even a 2020. So as you get more people involved in your mixture and you get more and more DNA information, it gets a little more complicated to kind of parse out each individual person's DNA types. So here, this highlights, it's a 2-person mixture. I see 4 peaks. I have no question that this is 2 people. We just talked about this one, so it could be, is it 2 people? Could it maybe be 3? Is there a lot of sharing? I'm not sure. And then a 3-person mixture, of course, we're going to add 2 more peaks to that location for maximum. So it could be anywhere from 0 to 6. And as you add more and more individuals, the number of max peaks per location also increases. So again, my beginning question, why are some comparisons sometimes complicated? And this is usually because of mixtures. So what if I show you something like this? This is a 2-person mixture. For me, this is relatively easy. It looks like I have a major and a minor contributor. Major contributor, the person that contributed more or the most DNA. So this individual, all of those really tall peaks, those I would expect to belong to that major contributor. The minor contributor are those shorter, smaller peaks. So on the second row down in the middle, D21, I have 4 peaks there. I have 2 semi-tall peaks and 2 short peaks. So this is pretty easy for me to know based on biology and that our peaks should be about the same height. Those 2 tall peaks go together and those 2 small peaks go together. And you can see that throughout the entire profile. But what if we have something like this? This is a 2-person mixture, 1 to 1, meaning each individual has contributed about the same amount of DNA. And as you can see in that top blue channel, a lot of the peaks on the right are all about the same height. So to the far right, TPOX, we call that TPOX. They're about the same height. So I don't know which 2 go to person 1 and which 2 peaks go to person 2. It could really be any combination, an 812, an 89, an 810, and so forth. And this is just continuation of this profile. So as you can see, all of these peaks are about the same height. So this is where it gets a little complicated. I can't tell which peaks belong to which individual. Now, what if I showed you something like this? So now I have way more than 4 peaks. This is actually a 5-person mixture, and this is what we would consider complex. As I go through each of these individual dye channels, all of the peaks are roughly about the same height. There is no way for me to tell which individual contributed which pieces of DNA. If you were to take my DNA and compare it to this, I may actually be in this profile just by coincidence because there's so many data points that I may happen to match them by chance. So as an analyst, this is where it gets a little complicated, not only to determine how many people are truly here. I never really know that. With DNA, I can say at a minimum it's 5. Could it be 6 because of sharing? Sure. But I know this is at least 5 people, but to be able to separate the individual people out into their own components is rather complicated. So all of these, what they all have in common is they're all mixtures, but as you can see, they're not all created equally. They range in complexity, they range in number of contributors, doses of different contributors. You throw relatedness in there, and that makes things a little more complicated. And then low-level samples with dropout. So do I have 2 people that have a lot of DNA, it seems, but as I move across the profile, one of those individuals might not have as much starting DNA as the other. So they're starting to drop out and I'm not seeing their data at all. But fortunately for us, we do have a tool to help with mixtures. This tool that we use is a probabilistic genotyping software called StarMix. We've used this for approximately the past 7 or 8 years, I believe now. And this really helps us with our um, mixture comparisons. So some of the benefits, we can now use data that we couldn't use before. So some of the previous ways that we perform calculations, we did a random match probability and a combined probability of inclusion. And with both of those, we couldn't use all of the data based on how tall the peak was. We do have thresholds that if we see a peak above a certain threshold, we can be confident that's the only peak we should see. Nothing has dropped out. In previous statistical calculations, sometimes we couldn't tell that. With probabilistic genotyping, we are able to take that into consideration. It also adds consistency to interpretation amongst myself and my coworkers, and it helps us determine potential contributors to DNA mixtures. So while it's not foolproof, it cannot tell us for certain there's 4 people in this mixture. There are some things that I will be showing you that help me understand, am I on the right track? Is it really 4 people or am I way off the mark and it should be more or less? So again, we use something as StarMix. There are other probabilistic genotyping softwares out there. This is what our laboratory uses. It assists us interpreting mixture profiles. And then it also helps us calculate that statistic, a likelihood ratio, which we will talk about in a minute. So for helping us interpret DNA mixtures, it uses biology and mathematical modeling to help us do something called deconvolution. And this is taking those individual contributors of the DNA and trying to parse them out into their individual components. So how does probabilistic genotyping software work? So again, step 1 is deconvolution. It uses those biological models to separate the mixture into its possible different contributors. So if you think I have a picture of a ball of yarn, if you think of a ball of yarn, it's all mixed up with all different colors. What StarMix is doing when I say it's deconvoluting is it's taking all the green pieces and putting them to the side. It's taking all of the red pieces and putting them to the side. Hopefully, so we can get a pretty good idea of which DNA types belong to which individual. It takes into account a couple different things. One is template. So this is how much DNA is there. Is one contributor contributing more than another? It takes a look at degradation. We know that DNA will degrade over time, especially larger loci. So those locations that are farther to the right when I showed the electropherogram, those are typically the ones that go first. And degradation, DNA breaking up, you can think of a long rope of DNA being cut into individual pieces. Also, amp efficiency. How well did the DNA copy? And is one person's DNA copying better than another? We do see that, and StarMix can help us determine that. And then we talked about stutter before, which is a byproduct of the DNA copying process. So it helps us parse out stutter peaks versus actual allelic DNA peaks. It uses something called the Markov chain Monte Carlo method, MCMC for short. This is used during deconvolution, and it's based on standard mathematical principles. It uses a sampling process, and it assigns a weight to each genotype combination. and then assigns a likelihood ratio for the profile given each explanation of the data. So you can think of it very similar. We like to say the hot cold game. We also like to explain when you go to the eye doctor and they're trying to determine which prescription is best for you, and they give you the choice of option A or option B, and you tell them B looks better, and then they give you the option of C or D, and you let them know C is better. They're trying to fine tune that prescription for your eyes. The MCMC process in Star Mix is very similar. So it's developing a DNA profile using math and biology. It's comparing it to what is actually seen in the profile, and then it's making adjustments to try to figure out, should these pieces belong to one contributor and these other pieces belong to another. Some common examples of MCMC that are used in the real world: engineering, search and rescue, weather predicting. So search and rescue, if you have, unfortunately, a boat that is capsized, search and rescue, what they will do is they will look in a 1 mile radius, and narrow that down as they get hotter or colder to where they think that that boat actually capsized, until they're in a pretty good space and they've narrowed it down to a pretty good area to search. That is the MCMC process. So the second thing that StarMix does is the likelihood ratio calculation. So it's using genotype weights and population frequencies that are well established and published, and it's calculating a likelihood ratio. For our laboratory, we report the statistic for the 4 main US population groups: Caucasian, African American, Southeast, and Southwest Hispanic. And then depending on where the case is from, we could add additional population groups. So for example, if we have a case that happened in St. Thomas in the Caribbean, we will add the Trinidadian population groups. So we also have groups for Native Americans, Guam, and the Northern Mariana Islands, as well as Native Alaskans. For Asians, we do not have an Asian population database currently. However, studies have shown that their population frequencies are going to be more in line with the 4 main populations of the United States. So it's not imperative right now that we do have an Asian population database. The database group is based on the evidence and not the population of the possible contributors. So even if I were told that a potential Chamorro individual is the suspect, if it took place in Manhattan, I'm only going to report the statistics for the 4 main population groups, not the Chamorro. Now, if it happened in Guam, regardless of the ethnicity of the suspect, I would then give the statistic for those population groups. So really it just depends, but it is all based on the evidence and not the individual contributor. So I've talked about a likelihood ratio several times now. What is it? A likelihood ratio compares the probability of the evidence given one of two different explanations, or what we call hypothesis. The first hypothesis is that a particular person of interest is a contributor. Generally, we call this the prosecution hypothesis. And then the second alternative hypothesis, generally we call this the defense hypothesis. And this is that an unknown, unrelated person is a contributor. And before I talk about this, so that using those two probabilities, actually I'll go back a slide. So using these two probabilities, whichever hypothesis has the higher probability, that is going to get the support. And the support, the magnitude of that support is determined by the two probability values. I do have hopefully a little bit easier of an example in just a slide or two. What is the likelihood ratio? So it is a numerical value. It ranges from 0 to very, very high number. The highest that I've seen for forensics is a non-nillion, which is 30 zeros. So anything in the middle there, the white grayish bar, anything that's a likelihood ratio of 1, we would say is uninformative. As you move from 1 to 0, it's a sliding scale. There's stronger support for hypothesis 2, or that an unknown individual is potentially a contributor. And then a likelihood ratio of 0 is an exclusion. Now, on the other side of that, the top part of this graph in green, the farther away that we get from 1 towards infinity, essentially, the stronger support for H1 or the prosecution hypothesis. And this is the words that I just said. So, all right, so hopefully a little bit easier of an example. I like to use this a lot in court. It tends to go pretty well. So let's suppose I have 2 bags of marbles. The first bag contains 10 marbles, 1 red and 9 blue. And I tell you that the second bag of marbles contains 100 marbles, 1 red and 99 blue. And I tell you that I've drawn a red marble, but I haven't told you which bag I've drawn from. So I can actually calculate a likelihood ratio. So for the 10 marble bag, I calculate the probability of selecting a red marble, which is 1 out of 10 or 0.1. And then I calculate a probability of selecting a red marble out of the 100 marble bag, which is 1 out of 100 or 0.01. Using these two numbers or these two probabilities, I can actually calculate a likelihood ratio of 10. So drawing a red marble is 10 times more likely if I drew from the 10 marble bag than if I drew from the 100 marble bag. Both are probable, but one scenario is more likely to the magnitude of 10. For DNA, we use much bigger numbers. So again, I can get into the millions, billions, and trillions. Verbal scale, we talked about this slightly before. So per SWGDAM guidelines, the Scientific Working Group on DNA Analysis Methods, I have to give a verbal equivalent in conjunction with the numerical likelihood ratio value. And this gives some kind of context to that number. I think telling a jury the likelihood ratio is 999,000, that doesn't mean a lot to people. But if I say it's strong support for inclusion as a contributor, I think that helps make that number give more meaning to that number and what that number actually means. This scale has been supported by the entire forensics community and was adopted by the Association of Forensic Science Providers and the European Network for Forensic Science Institutes. So now we're going to move, hopefully in the last 10 minutes or so, we can get through these next 2 practicals. So what I would like to do now is walk you through a profile, how I come to my conclusion, whether they're included or not, and I want to walk you through a star mix run. So again, we're going to go through this a lot quicker than what we can to really digest everything, but hopefully this will get you thinking. If you have any questions, my contact information is at the end. You're more than welcome to contact me or anyone in the DNA casework unit, especially if you are someone that's going to trial and need help, we certainly can help you. So for the first practical, I'm going to tell you that this is a 3-person mixture. We're not going to go through the entire profile for the sake of time. So I've just picked the first 2 or 3 dye channels. So this is the first channel. This is D3, or I'm sorry, it starts with D3. I know that this is a 3-person mixture based on looking at the entire profile and my interpretation. My interpretation is 3 people. I then have 2 POIs. One you can see on the right in green and one in blue. So first I'm going to take a look at the POI that's in green. And everywhere that I have a green arrow is where they're matching with that known individual to this evidence item. So as you can see for D3, I have 2 of the peaks that are matching, VWA, the 17 is matching, across the board. Those really tall peaks seem to align with POI 1. Now, if I compare POI 2, which is now the blue peaks, or I'm sorry, the blue arrows, at the first 2 locations, I see both of their DNA types represented, but as I move across the profile to the right, D16 and CSF, I don't have any blue arrows, and then I happen to see one at TPOX. So here, it's maybe they're there, maybe they're not. It could be that they match TPOX because the other individual also has TPOX. So then I will move on to the second channel. The second set of DNA STR locations in this profile, and I will do the same thing with POI 1 and POI 2. So POI 1, here, all of the green arrows, they are represented in every one of those STR locations. POI 2, it starts out pretty strong, but then as I move to the right, they have dropped out, or I'm not seeing those DNA types being represented. So now, what conclusions can I draw? So I do this across the entire profile, and determine, are there some consistencies? Are there not? So for POI one, the green individual, they're most certainly there. They align with the major contributor, the person that had the really tall peaks. They matched at all those locations. So I'm pretty confident that they are potentially included. For POI2, I'm not sure. I can't visually exclude them, so they do have some similarities, but I'm not really sure. So this is when I'm going to run star mix. And actually, I'm going to run star mix for both. In order to say someone is contributor, I do have to give a statistic. So I will run star mix. I use it a little more for POI2 to give me an idea of whether or not that person may or may not be there. So for POI1, this is a copy of what our star mix output looks like. It's very busy, so hopefully I can highlight the important things. So the first blue square that you see to the right, this lets me know when StarMix does its deconvolution. So it's taking that profile and it's separating out the peaks into its possible individual contributors. It gives me an idea of what that proportion of that first contributor is. So based on my knowledge and training and experience, I determined I thought POI was the major contributor, meaning they donated the most DNA. StarMix is agreeing with me. So they think that major contributor is about 89% of the entire profile. So that aligns with my interpretation. Then we have something called diagnostics. So that's the next section at the bottom. This lets me know how well the run went. So you can kind of think of this like a dashboard on your car. If there's a flashing yellow arrow or something on your dashboard, that may give you an indication that something is not quite right. We use this in a very similar way. The second blue rectangle, the Gelman-Rubin, that's how well the deconvolution went, how well the run went. Based on validation, we know that values should be about 1.2. Here it's 1.02, so that's within that acceptable range. As it gets higher, it may give me an indication that maybe my number of contributors is wrong, or something else was wrong with the run. Maybe the wrong sample was put into the system, or the wrong maybe a stutter peak, for example, didn't get crossed on or crossed off. Allele variance and stutter variance, those are also values that are determined during validation. The mode under each of those is about the average of where we're expecting it to be. So as long as it falls at or below those values, for good. It can be, for example, stutter variance. You see the mode is 13. We actually got an 18.9. That does not alarm me. Sometimes just in the DNA copying process, stutter may actually copy more than we expect it to. So it may be a little bit higher than what we're expecting, and that's shown in this run. So as an examiner, we train on this. We look at all different iterations of star mix runs to know what's considered a good run and maybe not so good of a run. The next page of the star mix output, it does calculate my likelihood ratio based on the individual population groups. So this particular example, I included not only the four main populations, but also the Trinidadian population. And we report the lowest to be conservative. So here that actually fell under the Trinidadian population at 3.3 septillion. When comparing that to the verbal scale, that would be a likelihood ratio that shows very strong support for the inclusion of person of interest 1. And this would be the numerical value that I report in my report. The next couple pages, just for your education, there is a per locus likelihood ratio that is calculated. So if for some reason, if I get an exclusion and I wasn't expecting an exclusion, I can come here, take a look at those locus specific likelihood ratios. It could be that I left in an artifact peak, a bacterial peak that shouldn't be there, and StarMix couldn't quite figure out what to do with it. So it may, looking at that likelihood ratio, it may give me some indication that something's wrong in my sample. So it does that for each location across each population group. And then here, this lets me know locus efficiency, so how well each particular section of DNA copied during the amplification process. We use this a lot. So for example, the number 6 at the bottom, that is the tallest peak. So the sixth loci, which I think is D8, if I'm remembering off the top of my head, that amplified the best. So that also gives me an indication if I go to D8 and look how many peaks are there, that may help me determine, knowing that amplified the best, that might help determine number of contributors. Whereas something like, for example, 9 that did not amp very well, I may only see 1 or 2 peaks there. That doesn't give me a good indication as to how many contributors are there. Parameters at the bottom, a lot of this is set during our validation studies. Again, it lets me know not only how is it running, how well it's running. So we take a look at that. And then the next 3 pages, this then, when it's deconvoluting, it's separating out into the individual potential contributors. It gives weight to each of the genotypes. So when it runs the first time, it's going to say, for example, at D3, a 1416. It compares it to the evidence that I input into StarMix. Yes, there's a 1416 there. It's going to accept that. Then it's going to change some other different parameter. Again, that hot, cold, is it A, is it B? And for each accepts, it does get a percentage of a weight. So for in this example, we have contributor 1, who is the major contributor. It really was able to pull out that contributor very well, and that would be my expectation. I visually could do that as an examiner, so I expect that StarMix would be able to do that as well. So almost every genotype combination or every combination of those DNA types at each location gets 100% weighting. So there was no uncertainty about whether or not that particular contributor had one DNA type over another. So I also take a look at this. If I have a major contributor, I'm expecting StarMix to be able to pull that major contributor. Here it did. So I'm confident that this run was appropriate. And this is a continuation. It does the genotype weighting for each contributor. As you can see, for contributor 3, the weightings are not 100% because it's not really sure. It could be a 1717. It could be a 1517. Each time that knob was turned hot or cold, a 1717 one time was a pretty good fit, but then the next time a 1517 was a good fit. So as you get further down in smaller amount contributors, you could get a little more of this uncertainty and the weighting spreading out over different genotypes. Exactly what I would expect. And then the final couple pages of the StarMix run, it gives, it lets you know what your evidence profile was. That's the middle pane. It lets you know what were the genotypes and the peak heights that you put in. If something's wrong, I can go here to make sure there wasn't a random peak, a bacterial peak, for example, or a peak that shouldn't be there. So I compare this to my evidence profile to make sure that the file that I put into the system is what I'm expecting it to be. And then to the far right, this is the reference sample profile. So POI1, that green POI, this is all of their DNA types. This is the file that I put into StarMix. Again, so I want to make sure that this is exactly what I have as well. It could be that you pulled the wrong file. You want to verify to make sure that it actually is the right file. One thing I didn't mention before with StarMix, so it does the deconvolution process, that MCMC process, and the genotype weighting before it does any comparisons to a reference sample. So we actually don't need a reference. In cases where we may not have a reference and we're trying to determine a profile for CODIS, for example, we can run it without a reference and it will deconvolute the sample. It will give us an expectation of the different DNA types for the different contributors that I can then use. So the reference input file does not have anything to do with how it deconvolutes, with one exception. If I'm conditioning on someone, so that meaning if I have a vaginal swab from a victim, I'm expecting her DNA to be there. So I'm going to tell StarMix, I know she's there. There's no question that she's there. it's going to take that into consideration when it's doing its deconvolution process. It's going to say, well, I know the victim is a 1414, for example. What else do I have here? Maybe that second contributor is the 1516. So that is the only time during the deconvolution process that a known sample actually is relevant. Otherwise, it does all the deconvolution first, and then it calculates the likelihood ratio based on population frequencies and comparing to that POI. So for the second POI, I do the same thing. So actually, I run something called an LR from previous. This is the same deconvolution that happened the first time, but now I just want to calculate a statistic based on that second POI. So here you see the same page. Under summary of contributors, POI2 aligned best with contributor 3, which before I think we saw was like a 2 or a 5% contributor, which was my expectation. As we put arrows on that electropharogram, I wasn't really sure. They weren't really there really, really well. So that would be my expectation, a low-level contributor. And StarMix is aligning that person as well with my interpretation. So for this one, again, we run the same statistics. Trinidadian came in at the lowest. So the likelihood ratio is actually 1 over 380. So this would be an exclusion. Anything over 1 over 100 or 1.0 times 10 to the negative third, I believe, or -2, -3. It is an exclusion. So in this situation, I could report the individual as excluded. However, I couldn't do that visually, but based on star mix and population frequencies and how the sample was separated out into its individual contributors, there was support for exclusion of that individual. I'm going to run through number 4 really, really quickly. I do the same exact thing as I did for our 3-person mixture. So here again, I have 2 POIs, a green one and a blue one. Here are all the green peaks that align with POI 1. As you can see with this sample, it's not as clear as the other one. There isn't really a major contributor. You'll see that more as I go on. The second POI, the blue one, about the same amount of arrows as the green. I move on to the next location. Kind of the same thing. Now remember, this is 4 people. So even though, for example, at D8, the 14 is really high, it could be that 14 is made up of 3 different people. So I can't really call that a major contributor knowing that I'm expecting at max 8 alleles here and I only see 3. So there's probably some sharing going on. There's probably some dropout going on. And then for this one, I actually did want to show you a third channel because this looks very crazy to me. So here we're starting to get a lot more information. It's relatively the same height. I really can't tell you which piece of the puzzle goes to which individual. Green individual. So for D2, the first location on the left, I see both of their peaks. For D19, the next location over, they don't match with any of those peaks. But then they do for the next 2, THO1 and FGA. So are they there? Maybe, I'm not really sure. And the same thing with the blue. So before I had a lot of arrows, now we're kind of limiting the number of arrows that actually match. So again, I do this across the entire profile and I ask myself, is POI1 included? I don't know. Maybe they are. I can't visually exclude. I'm not sure. Same thing with POI2. I run star mix. Here's my star mix run again. For POI1, it aligns it with the number 4 contributor, the 4% contributor. Based on what I see, that makes sense. I didn't see arrows at every location. There were some locations where I didn't see an arrow at all, which would make me think that that low-level contributor is not there a lot. The statistic for that run is actually 1 over 2. So 0.5, we would designate this limited support for exclusion. So we can't outright exclude, but there is support that POI1 is potentially excluded as a contributor. For POI2, same thing. It actually aligns with that low-level contributor as well. That makes sense. If you have a low-level contributor, it's possible that multiple people can align with that contributor. Again, going back to my phone number example, we may only have equivalent of the area code represented, and maybe both of those POIs have the same area code. So that does not cause alarm for me that that second person, based on how many arrows I saw and how many consistencies between the DNA profile and the evidence and the profile from the known person, that makes sense that I'm not sure that they're there. So the fact that they align with the lower contributor makes sense. The likelihood ratio for this is actually 1 over 2,577, which is well over 1 over 100. So we call this an exclusion. So lastly, what happens if we get it wrong? So forensic DNA is never exact. We use our training and experience to determine the best number of contributors based on the data that we see. We use star mix sometimes to let us know if we're not sure. It might be 3. Star mix might let us know potentially it's 4 based on some of those diagnostics that we talked about. Our developmental and our internal validations have tested what happens when we get the number of contributors wrong. For both of those validations, we actually create our own mixtures. So they take a part of my DNA and they take a part of my husband's DNA, and they know that that should be a 2-person mixture. But when they actually perform DNA testing, maybe they put a cup of my DNA and a teaspoon of my husband's together. So we know maybe my husband won't show at all. Or maybe if it's a cup and a cup, maybe we'll see all of both his and my DNA types, but we can't tell which one belongs to who. So in validation, we can control that uncertainty. So in doing that, we could see what happens if we know it's a 2-person mixture and we say it's 1, or we know it's a 2-person mixture and we say that it's 3. So typically, underestimating the number of contributors leads typically to false exclusions of true contributors. So if we have a 3-person mixture that we say is 2, and we have that really low level third person, they're probably going to be excluded falsely. Overestimating the number of contributors could lead to false inclusions of non-contributors. So again, if I take a true mixture of 3 people and I say that it's 4, I can then maybe falsely include some individuals that aren't really there because I can have them match by chance. For the major contributor, there's not much effect. So that very first example that we had, the major contributor, the peaks were really, really high. There was no question that those peaks go together. If it's truly 2 and we say it's 3, that first tall major contributor is always going to be contributor number 1, more than likely. So there isn't going to be much effect. The minor contributor is where we see the greatest amount of effect. So that low level contributor, again, if I know that it's 2, but I say that it's 3, we're going to get a low likelihood ratio. Now, I'm not going to get a likelihood ratio of 1,000,000,000. I might get a likelihood ratio of 2 or 3, just because they happen to match by chance. But that's why we give that verbal scale too. We have that range. A likelihood ratio of 2 or 3 is not super informative versus a likelihood ratio of maybe 1,000,000,000 or a non-million. We use those diagnostics that we talk about to help us determine, are we interpreting the profile the best that we can? It helps me determine if something went wrong. For example, I have the wrong number of contributors. Sometimes I say it's 3, but I accidentally type a 2. I use those diagnostics to help me understand that. I review the data, everything that I showed you today, as well as the input files, to make sure that really the profile I thought I was interpreting is the one that I truly input into StarMix. So that was a lot. I really appreciate you hanging on with me for the past hour or so. Hopefully that gives a little more insight into mixtures and why they can be complicated and probabilistic genotyping. Certainly, if you're a practitioner that's going to go to court, work with your witness. We can walk you through the statistics and make it a little more palatable in the courtroom. If anyone ever has any questions, feel free. My contact information is here, my email and my phone number. Feel free to give me a call. Certainly, this is something that we would need a week-long training to really get into the nitty-gritty, but hopefully it was a good start. And certainly, if there are ever any questions, please don't hesitate to reach out. So I thank you, Jasmine, and I thank you, Dr. DeZuno, for having me today. I don't know if there's any questions from the audience. If so, I'd be more than happy to take them. If not, thank you so much.
Amanda, this is Joe DeZino. It doesn't look like there's any question.